The broadcast is now starting. All attendees are in listen-only mode.
Hi, good afternoon, everyone. This is George Orling from HDSA, and I want to thank you for joining us for the February installment of the Research Webinar Series at HDSA. I thought I'd take the opportunity today to just kind of use the time to walk through and review the progress that not only that was made here at HDSA as it relates to novel drugs for HD and research and breakthroughs that have been made, but also around the globe and kind of give a high-level overview of the progress that's been made and what we've learned in 2016 as we, the research community, continue our hunt for effective therapies for HD. So, most of you are I'm sure very aware of how this program works, the GoToWebinar program. But just in case there are folks on the line that are new, you can ask your questions at any point during the webinar. Just type in your questions in the box listed. You should see a menu box on the right that says questions. You can type that in, and at the end we'll go through them and try to get to as many. I'll try to answer as many questions as we can, or that I am aware of the answer of. We are recording this, so in case you have to dial off or you know folks that would like to hear it, not a problem. We will record this and archive it both on our website as well as on our YouTube channel, which can be easily found by going to hdsa.org and clicking on the middle icon here. It's hard to see, but the YouTube icon here, and it'll take you to all of the different videos and things that HDSA has produced over the past few years. So maybe you're interested in this talk, or maybe you're interested in hearing more about ASOs or gene Huntington lowering technologies. We have webinars about all of those different topics, so check out the YouTube channel. So I just want to start by, no matter whether we're HD patients, caregivers, family members, scientists, or just general community members, we all share one common goal, and that's we want new drugs today for Huntington's disease. But we know that they just won't fall from the sky. We've got to be more proactive and strategic in our planning to ensure that we meet our goal of finding new effective therapies for Huntington's disease. And I think we need to develop, and what I'll talk to you about today are 4 different things in the pipeline, and this isn't just HDSA's pipeline, this is just a general HD research community pipeline that we need to ensure is full to develop these therapies. First is, obviously, and first and foremost, is drugs. We need a robust drug pipeline that involves not only small molecules and antibodies and Huntington lowering drugs like antisense oligonucleotides, which we'll talk about, but also drug targets. We need new targets. Besides Huntington, what else can we, as drug hunters, develop or identify as involved in, maybe potentially being involved in HD, that we could develop a drug to target and potentially intervene in HD? If we have the drugs, we also need the clinics. We need the clinics not only to provide excellent care for HD families and patients, but also to be the ones that are carrying out the clinical trials that will facilitate the development of these drugs in the pipeline. Inside those clinics and inside the laboratories, we need to ensure that the pipeline of young researchers and clinicians is full for the next generation of HD families. And finally, no matter what drug we have or how fantastic the clinic is and the scientists and the clinicians are that staff that clinic, we will not have a drug if we don't have research participation from the HD community. So we'll talk a little bit about that and the things that are going on globally to help facilitate that. So I just want to start just with 1 or 2 slides about what I think is very exciting in terms of HD research in the past couple years. This is a slide that many of you have seen if you've gone to an education day or heard another doctor speak about what HD is. This is something we know as fact, and that is that, as hopefully you can see, that on this right-hand graph, that as the number of Huntington is caused by a polyglutamine repeat expansion, and when you have 40 or more Huntington repeats, if you live long enough, you will develop the symptoms of the disease. That's known, and what we also know is that as that CAG repeat expands, you will develop the symptoms at an earlier age. So there's an inverse correlation between the number of CAG repeats and the age of neurologic onset. And what I mean by age of neurologic onset here is this is a motoric symptom. These data were generated by determining someone's motor onset. While we know this to be true, what you should also notice is this pink coloring here. There's a lot of noise in these data. So CAG expansion can only account for 50 to 60% of the age of neurologic onset. There are a lot of this pink coloring indicates there's a lot of other variables likely at play that could be causing, why someone, for instance, at the age of less than 20 with around 40 CAG repeats develops neurologic onset of the disease here, but someone with 40 CAG repeats can also develop HD up near 75 or 80. What is it? Why is that happening? What is it about this person at 75 or 80 years of age with just 40 CAG repeats that seems to be maybe protecting them from the disease? Or on the converse, what is it about this 20-year-old that is causing them to have an age of onset that's younger than we would have predicted based on their CAG repeats? This is really exciting, is trying to figure out why we see this. And in 2015, we talked about a landmark paper that came out, a GEM HD consortium paper that had hundreds of HD researchers on it that did something called the GWAS, a genome-wide association study, where they're starting to look for the hunt for genetic modifiers of Huntington's disease, genes that could be causing that pink signal that I just showed you in the last slide. And there was an exciting paper, I thought, that came out from Leslie Jones's group in Cardiff University in the UK from Betancourt. It appeared in the Annals of Neurology late last year. And what they're now doing is starting to narrow down within different regions of our chromosomes where there may be some potential genes or genetic modifiers of the disease. In the 2015 paper, they kind of identified a couple regions in chromosomes 15 and 8 that seem to be kind of like the neighborhood, within that neighborhood of those chromosomes, certain neighborhood within the chromosomes, there appeared to be some hot spots where potential genetic modifiers may be. And this paper, while it isn't definitive, it's definitely has shown that there are a couple genes, FAN1 and PMS2, that are basically, this paper showed that there's a significant association with genetic variants of these two genes in the age of onset of Huntington's disease, as well as other polyglutamine diseases like SCA1, spinal cerebellar atrophy. Why is this exciting? It's because this is target validation. These are potential new targets that could open up a whole new HD drug discovery programs. And it's exciting because they're data coming from humans. These data weren't identified from the Betancourt paper from a mouse or a fly model of Huntington's. These were pulled out of human genetic data. So they're real, and while there's a lot of work remains to be done, it's this type of approach that is very exciting and could lead to potential new therapies down the line. And what's also interesting about this paper and what was found in the earlier 2015 GWAS paper was that both of these genes, FAN1 and PMS2, seem to be involved with DNA, the way your body handles DNA repair or damage. Our body is really beautifully set up so that when there's some sort of DNA damage or some mistake that your body makes when replicating DNA, it can be fixed. But sometimes, and we believe in Huntington's disease, that goes wrong somehow because that is how the CAG gets expanded. And it's interesting to find that these two genes and potentially others may be involved in the disease process of Huntington's disease. So this is super exciting. So I would say watch this space around DNA repair and damage for Huntington's disease. I think we'll be hearing a lot more of that in the years to come. So that's all I want to talk about in terms of new targets for Huntington's disease. There'll be more coming, I'm sure, from the exciting work from the genome modifiers work that's being done by researchers all around the world. I next want to just talk about within HDSA, provide a brief update about our HDSA Centers of Excellence program. This has historically been, a place where you can find your expert multidisciplinary care for your Huntington's disease. But we want to make sure that the Centers of Excellence are not only providing great HD care, but also working towards an HD cure. In 2016, we've seen our centers grow from 29 centers in 2015 to 39 Centers of Excellence in 2016. That number incorporated 11 new Centers of Excellence, and HDSA committed over $1 million towards this exciting program. Not to go over the dates here, but this is an annual program that we review annually here at HDSA with the help of experts and volunteers from around the country that help evaluate the Centers of Excellence. And we expect that 2017 awards for centers will be announced in early February of this year. This is just to show you how far we've come, but that we still have work to do. When the HDSA Center of Excellence program was not rebranded, but did a reboot and became a more competitive and annual renewal program, we had 20 Centers of Excellence in 2014 covering 17 states shown in blue. And as of today, we have 39 Centers of Excellence in 30 states shown here. I mentioned the research and clinician pipeline. That's something we take very seriously here at HDSA, and we think that it's essential that we pay great attention to that, because the researchers and clinicians at these centers and labs around the world aren't getting any younger, and we need to make sure that the next generation has folks fighting on their behalf. One of the programs that we run every year, and is one of my personal favorites, is the Donald King Summer Research Fellowship. And this past year, we've awarded 3 fellowships to Lance Heedy, Danny Berge, and Zhuguan Zhang, shown here, to work in laboratories around the country, spend their summer working with HD experts on really exciting projects. These are not your normal high school science projects. These are really detailed science projects working with HD experts that are moving the field forward. And we're happy to have them, and look forward to having, you can look forward to having a chance to interact with these fellows as well as other HDSA fellows at our convention in June. Our largest research project that started in 2013 is continuing and moving along very well. It's called the HD Human Biology Project. This is a grant program that's open to researchers and clinicians all around the world to fund them to do one thing, and that is to help us better understand the biology of Huntington's disease as it occurs in humans. We have made a very strategic decision to place our resources where we think it matters most, and that is to understand HD in the only organism that gets HD, and that's humans. So this program we launched in 2013, and since then we've awarded over $3 million to researchers around the world. This past year, just a few months ago, we awarded 9 fellowships to the researchers you see here on your screen. I won't go through all of them, and all of the information and summaries about these exciting research is on the HDSA website. But it's really exciting work that runs a gamut from looking here at Indiana University, looking at transcranial direct stimulation for as a invasive way to improve some of the symptoms associated with HD, to basic research biology at the University of California, San Diego, where we're looking to identify the protein interactome, all of the different proteins that interact with the Huntington protein in juvenile HD brains. This could also open up a new, better understanding of the biology of juvenile HD. So a lot of really exciting work that's being funded from studies directly in patients, as well as using human material to better understand the biology. We've expanded our research portfolio in 2016 to kind of keeping with that theme of making sure the pipeline of researchers is full by the launching of the Berman Topper Family HD Career Development Fellowship. And this year we're really excited to announce that the winner is Dr. Ricardo Moro-Pinto from Mass General Hospital, Harvard Medical School. And Dr. Pinto is working on using some exciting techniques you may have heard of, like CRISPR, that is a technique that's used in genome editing, to use this to help validate some exciting new targets that are coming from the genetic modifying or the genome-wide association studies that were published last year that I alluded to a couple minutes ago. Why are all of these important? So, you know, the work that we do at HDSA, as well as other research foundations dedicated to HD, are so important because, you know, unfortunately, the research funding for HD has decreased over just 4 years. And this is data directly from the NIH on money spent in millions towards basic HD research. And in 2012, there was approximately $65 million dedicated to HD basic biology research from the National Institutes of Health. And while these are estimated values, the end of 2015 was 39 million, and they don't expect, at least they're not estimating that it should not increase over the next 2 years, 2016 and 2017. So a 40% decrease is not moving in the right direction. It places a strong emphasis or rationale for why we must continue to do what we do and support young researchers in their endeavors. So last year I showed a similar slide like this, and 2015 was a great year at the FDA for new molecular entities, or NMEs. These are basically drugs that are approved by the FDA for an indication like Huntington's disease. And last year it was one of the best years on record, where they had 45 different new drugs approved from the FDA. Unfortunately, we had a huge decrease in 2016 with just 22 drugs approved from the FDA. Now, there are reasons for that huge discrepancy. That some will argue that, there were a few that were not approved that we were hoping to be approved, but there were also some. There were also some that were approved in 2015 that were originally not expected to be approved until 2016. So while it can account for some of this decrease in approvals, it certainly can't account for all of it. The good news still here, if we're to find there's two things of good news here, is that of the 22 drugs that were approved, 41% of them were approved to treat rare or orphan diseases like Huntington's disease. And additionally, this blue line here, these are applications or filings. These are these were filed in 2016. These are kind of in the queue for approval in 2017. So we didn't see, while we saw a decrease in approvals, we did not see a decrease. We actually saw an increase over the past year in the number of drugs that were submitted to the FDA for approval. So I suspect we're going to see a nice rebound in 2017. This is just to show you that there's working in the rare disease space, there's reason for optimism. I would just draw your attention to the bars on the right-hand side, where these are data of clinical trial success rates for different diseases. This is in blue is all disease, in the yellow is chronic high prevalence diseases like cancer or diabetes, and then in the green are rare diseases like Huntington's disease. And what you'll see is this is a calculation of the % chance of approval for a disease or drug for a particular disease, going from phase 1, which is the very first phase of clinical development, all the way to approval from the FDA, which is what we want. And you'll see that for chronic high prevalence diseases like Alzheimer's and diabetes, you have a success rate of under 9%. But for the rare diseases where we have a better understanding in many cases of the biology underlying the disease, like HD, we have nearly a 3-fold increase, 25% chance of approval. That's really impressive and reason to be optimistic for the new drugs that are being submitted to the FDA for Huntington's disease in the future. So this has got an updated pipeline for 2017, not of course inclusive of all the different things that many different pharmaceutical and biotech companies are working on the left-hand side. As we all know, there's just one drug approved by the FDA for this is tetrabenazine or xenazine for the chorea associated with Huntington's disease. And what I'll do, and I have updates here of where some of these drugs stand in the pipeline. We'll be going through, I'll be touching on some brief updates on pretty much the ones here shown in different colors, like the Pride HD, Legato, the Pfizer study, the Vasinex study, the Azavan study, and a couple others. In 2016, I think we were all kind of hoping that we'd have a new drug on the market approved for Huntington's disease symptom management. This is a drug that was part of a clinical trial called First HD and ARC HD from Auspex and then Teva Pharmaceuticals. This drug is SD809. It's basically an analog version of xenazine. It's basically, which is shown here. They just take some deuterium, which is kind of like heavy hydrogen, and they made a simple modification of xenazine, and what they found was that the drug, SD809, had a very significant improvement in total maximal chorea score in HD patients, and it also had an improved side effect profile compared to xenazine. So these data were very exciting, but unfortunately, the drug, the original application as it was submitted in 2016 was not approvable by the FDA. They requested additional information, and from my understanding, that information has been provided to the FDA, and we hope to have a decision we hope to hear a decision from the FDA on this drug, SD809, by April 3rd of this year. So stay tuned. So the pipeline, that's an updated, that drug and that trial is complete, and we're just waiting to hear from the FDA. But there are a number of other drugs that are in development, either currently recruiting or just wrapping up, or in the planning stages for clinical trials. But in terms of the pipeline, I see the drug development pipeline starting with Enroll HD. Enroll HD is a global, it's many things. It's a global patient observational study, longitudinal observation study that is tracking patients, caregivers, and family members over time about the effects of Huntington's disease. It has 3 major goals. One is it wants to help us improve our understanding of HD and kind of get at those questions we talked about in the early slides about what factors influence the progression of the disease. By tracking thousands and thousands of HD patients across many countries and continents, we'll also hopefully be able to foster good clinical care and understand why some patients may benefit or do better in a certain country than another. By maybe it's something the way the doctors treat them. By having data over thousands of patients, we may be able to extrapolate some exciting information that could improve the clinical care of patients. But I think the real power of Enroll HD is to enhance and design and expedite the conduct of clinical trials. When the next clinical trials or companies are ready for their drugs to go into the clinic for HD, they're going to want to know where the patients are. And they're going to turn to something like this Enroll HD, this platform, to launch their studies. So if you have an Enroll clinic or site in your neighborhood at your particular HD clinic, and it's not well populated with HD family members and patients, the likelihood of a pharmaceutical company selecting that site, I would think, would be very low. So it's important for us as the HD community to make sure that if we do have an Enroll site, we consider participation, because one, it's moving the field along, but at the end of the day, we want a clinical trial in our neighborhood. We don't want to have to go, a couple hundred miles once a month to participate in a study. It would be great if it was in our local HD clinic. So as of January 1 of this year, to give you an idea of the scope and the growth of this program, there are almost 13,000 participants in the Enroll HD study across 14 different countries. This year, the Enroll HD released a new data set, which is exciting for for researchers. This data set includes detailed clinical data on nearly 9,000 participants. And what's great about this is it's freely available to the HD research community on the Enroll HD website. So if people are interested and have a question, they want to tap into these data, they can by contacting the folks at CHDI or Enroll HD. Below you'll see who are, and hopefully you can read these, who are the top enrolling sites for the past year. And hopefully what you'll take from this is that there is just one American site, the Hereditary Neurologic Disease Center in Wichita, Kansas, that has cracked the top, say, 15 in the past year in terms of recruiting. So while we do have quite a few patients in Enroll HD, participation in U.S. sites is lagging far behind Europe. So let's start with just to give you a brief update on, I think, the study that family members and patients know a lot about and have heard a lot about. This is a study being sponsored by Ionus, formerly Isis Pharmaceuticals, in partnership with Roche Pharmaceuticals. And this is a study to investigate an antisense oligonucleotide, or ASO, for HD. Now this study is a very small study. It's a phase 1B, which is it's a safety study in HD patients that began slowly in late 2015. And it's important to note that the purpose of the study is simply to assess the safety and tolerability of this drug, Ionus Huntington RX. We are not going to learn whether or not this drug is efficacious or works to delay the onset or the progression of Huntington's disease. It's not designed to do that. That will happen in future studies, we hope. How this drug works is that this drug is not like a typical pill that you would take orally, like an aspirin. It's actually like a piece of DNA shown here. It's like a helical structure. You can't take it orally. It has to be actually administered via a lumbar puncture or a spinal tap, which we call intrathecally. And when it's administered, it lasts for a long time. It has a duration of action about 4 months. So it's not something you have to do every week or necessarily even every month. The idea is hopefully that maybe once a quarter you would have a administration of this drug. What the drug does is it binds to, this particular drug binds to the messenger RNA of the Huntington gene. And it binds to both the mutant or the expanded Huntington gene, the one with 40 CAGs or more, and it also binds to the messenger RNA of the normal or wild type or non-expanded Huntington gene, that that normally has around 20 CAG repeats. So it can't discriminate between the mutant and the wild type or the normal, so it's not what we would call, it's non-allele specific. that could be just fine. We don't know. That's why we do this study. But the end result of this drug is what it'll bind to the messenger RNA of Huntington. Your body will recognize this as foreign. It recruits a bunch of little enzymes that come and chop this thing up. And when you have less of this mRNA, which is basically a recipe for protein, you'll have less of the Huntington protein. So the idea and the hypothesis is that if we can decrease the amount of Huntington protein in the brains of HD patients, we could slow or halt disease progression. So this study, unfortunately, is not going on in the United States. It's at sites within the UK, Germany, and Canada only, and just recruiting a small number of patients, around 36 to 40 some odd patients around the globe. The good news is it's 2017, and I can say that this study is targeted for completion this year. And as of the latest update, the fourth and final dose of this drug has been given. So the way this trial has been working is they started with a very low dose to make sure it was safe and not toxic to patients, and they've been slowly and incrementally increasing the dose over time, and now the fourth and highest dose is being administered to patients as we speak. So as I said, the study is targeted for completion around September, and we hope to have results shortly thereafter. While you may have missed it, this was something I thought was incredibly exciting and reason for hope and optimism in the HD community and other neurologic disease communities. Ionus has a drug, another antisense oligonucleotide drug called Ionus Nursin, that was, or Ionus SMN-RX, that was in the clinic in collaboration with, I believe it was Biogen, in a clinical study for what's called SMA, spinal muscular atrophy, in an infantile onset, or early onset SMA, which is a really it's a genetic disease. It's similar in nature to HD, and very aggressive, where these children with infantile onset never really develop the ability to stand up or walk and have difficulty breathing and die very early. What they've done with this drug in the study called the Phase 3 ENDEAR study was they developed an ASO that doesn't actually decrease the amount of a gene like the Huntington ASO is designed to do, to decrease Huntington. This ASO is actually designed to increase the expression of a gene called SMN2. And the idea and the hope being that if they can increase the expression of this gene, they could restore function in in SMA patients. It's an ASO. It works the same way as the Huntington ASO does. It's delivered the same way as the Huntington ASO is via spinal tap. And they found that this drug has been in over 250 patients over 4 years. It was safe and effective, and the study, the ENDEAR study, was actually stopped early. And in the Huntington's disease community, when we hear drugs are stopped early, they're usually terminated. They've been terminated for futility, meaning they didn't work. Well, this study was stopped early because the interim analysis of the data suggested it was so effective that it was, they had to stop it to get this drug to the FDA for approval. And it has been, as of, I think it was December 23rd of this year, this trial stopped and the drug was approved by the FDA at the very end of the year. I put this up here to talk about the pipeline a little bit more, and I won't go into detail of all of the different companies and drugs that are working on this in this space, but this is a depiction of the central dogma of molecular biology. This is, for those molecular biologists or Biology 101 people out there that have took maybe biology in college, and maybe this is the one thing you had drilled into your head, is that DNA makes RNA and RNA makes protein. This is kind of the recipe of life. And another way to look at it, and I look at it, is just simply that DNA is our cookbook. Inside that cookbook, there's lots of different pages and lots of different codes and instructions And inside that cookbook on a particular page is a recipe, and the recipe is really your RNA or your messenger RNA. And by reading your recipe and executing the protocol on the recipe, what you make is the protein or the meal. And what we've seen over the past, I'd say 5 to 10 years, is a transition to a strong emphasis on this kind of central dogma around Huntington's disease. Huntington's disease has a great benefit, not a benefit, but a strength in that it's a monogenetic disease. We know what causes this disease. It's the expansion of the CAG repeat in the Huntington gene. Other diseases like diabetes and Alzheimer's and Parkinson's don't have that luxury. They're still searching for the causative genes of these diseases. We know what causes the disease, so now that we have the technology that can target the molecular, the DNA, the RNA, and the protein, a strong emphasis over the past few years has been placed on using different modalities like zinc finger proteins and CRISPR, which is coming in the future, and small molecules to target DNA. We just talked about ASOs, and what's coming are things using viruses to deliver silencing reagents or shRNAs to the brains of HD patients. And then there's even ideas, and folks are working on ways to, for instance, wouldn't it be great if we could develop a small molecule that could trigger our body to induce something called autophagy or self-eating that would cause the body to chop up and digest or degrade the Huntington protein preferentially, and that could potentially be a disease-modifying drug. So lots of different companies are working in this space, and you'll be hearing about them both at HCSA convention as well as in the coming years as they move closer and closer to the clinic. But it's just up there to show you that there's a lot of great things going on in the background that we're not going to talk about today that we hope will see the clinic someday soon. While that's going on, you know, it's great, and we do believe and think and hypothesize that lowering Huntington, it makes sense, right? It's just common sense. If we can decrease the levels of Huntington in the brain, it makes sense that potentially this could be a disease-modifying approach. There's still a lot of things we don't know. So what's happening in the background, and this could occupy, each one of these bullets could occupy several hours of research webinars. Really laborious, boring kind of stuff, but it's really essential that we answer these questions. We need to know, and still don't have the answers to, you know, if we do have a Huntington lowering approach or drug, where do we put it? Do we have to put it, inject it directly into the brain? What if we can only lower Huntington in the periphery, in the muscle? Will that be okay? We don't know. Who do we have to treat? And when? At what point during the disease progression is it too late, or is it ever too late? Can we take someone who's late stage HD and have them restore function? We don't know that answer. How much Huntington do we have to lower to have an effect in a person? We have some ideas in mice and monkeys, but mice and monkeys aren't people, as you all know, and the translation of those findings in mice is usually not very good in people. So we need to better understand how much Huntington do we have to lower to see an effect. We talked about the Ionus approach, where it's a non-allele selective approach to lower Huntington. So this drug will lower both the wild type or the normal Huntington and the expanded Huntington. There are companies working on an allele selective approach, so that would we just lower the mutant Huntington. Is that preferential over a non-allele selective approach? We don't know, but studies like the Ionus study and future studies will be really instrumental in helping us to understand that. So far, all of the approaches like the Ionus study and some of the gene therapy studies that are going to be are moving forward, they involve intrathecal injection or will involve a direct injection of a virus directly into the brain, like the striatum of an HD patient. It's not ideal, and wouldn't it be great if we could develop an oral or intravenous drug that could serve the same purpose, and we're working on that as well. At the end of, so I just want to walk through some of the progress and specific HD clinical studies that have happened over the past 12 months. There was a study called the PRIDE HD study. This is a study sponsored by Teva Pharmaceuticals, and it was a large study with around 400 subjects studying the effect of pridopidine, which goes by lots of different names you may have heard of, called as Huntexal or ACR16. This study was looking to see if the drug pridopidine would improve the total motor score, TMS, in HD patients. And we've seen this before. This drug has been in the clinic in 2 studies called HEART and MERMAID that while total motor score was not the primary outcome that was declared by the developers for these HEART and MERMAID studies, they did see a significant improvement in total motor score in those 2 studies. So this study was looking to, originally designed to see if the drug could improve total motor score in HD patients. Recruitment was completed, and the initial results were announced in September of last year. And unfortunately, what the results showed was that there was no significant improvement in total motor score at any of the doses given. However, there was a significant improvement in something called total functional capacity, or TFC, at the low doses, BID, which is means twice a day. Total functional score is just a clinical assessment that's shown here on the right. It kind of tries to take into account 5 different areas of normal day functioning and to quantitate how a patient is doing in handling their occupation, their finances, their domestic chores, their ADLs or activities of daily living, and their care level. Someone who scores a 0 is not doing well and is in more late stage HD, but someone who scores at a 13 is fully functioning and has probably no overt signs of Huntington's disease. What they also found in analyzing the PRIDE HD results were that the greatest effect was seen in the earlier stage 1 and 2 HD patients. So what's next? Well, I can't say for sure, but likely based on the results, would be a phase 3 clinical study in early HD patients, because that's where they saw the greatest effect, looking to see if the drug can improve total functional capacity. Late last year, another drug study called the Amaryllis study, sponsored by Pfizer, announced their results. And if you want a really great summary of the findings, can be found at hdbuzz.net. They have a nice article about that appeared sometime I think in December of last year. This study was studying an inhibitor of an enzyme called phosphodiesterase 10, or PDE10. This is a protein that's found in the striatum of human brains, and you can see it here in a healthy brain. This is a PET scan of a healthy human being, and these kind of pea-like structures or kidney-like structures here, that's your striatum, lit up green and yellow. That's indicating a high level of expression of PDE10. And what we know and have observed in both animal models as well as HD patients is that as the disease progresses, there seems to be a decrease in the levels of this enzyme in HD brains. The hypothesis was that this might be a good thing, that these levels are decreasing, and that an inhibition of this enzyme could ameliorate both motor function as well as potentially improve some cognition and behavior endpoints. Unfortunately, the data were analyzed, the study was completed, and it failed to meet any clinical endpoints. Teva has a second study in the clinic. It's still recruiting. It's called Legato HD. This is basically a a study testing laquinimod, which is an anti-inflammatory drug for the brain. And they're now testing the effects of 2 different doses of this drug on total motor scores, similar to what we saw in the PRIDE HD study that I just explained. This study started a couple years ago, kind of went on a little hold because it was found in another clinical study, I believe it was for multiple sclerosis, that the higher doses had potential cardiovascular side effects. So this dose, the higher dose of 1.5 milligrams per day, was dropped from protocol, and protocol was amended, and now this study is up and running and recruiting again. One of the only other studies that's currently recruiting in America is called this NeuroNext trial sponsored by the NIH is called STARE. STARE stands for, their drug is SRX246, and it stands for Safety, Tolerability, and Activity in Irritable Subjects with HD. This is a drug it's called an antagonist of a vasopressin 1A receptor, and the idea is that this receptor is somehow involved in irritability and aggression, that if you can inhibit this receptor, you could improve some irritability and aggression symptoms in HD patients. So this study has started testing in early symptomatic HD patients. It actually is recruiting in 22 U.S. sites around the country. Started in the middle of last year and is still recruiting. One final study is called the Signal study, and this is was a phase 2 research study testing people who are at risk and early signs of HD, testing a drug called VX-15, which is an antibody. It's not a small molecule or an antisense oligonucleotide, but it's injecting an antibody that recognizes this protein called semaphore 4D, or SEMA4D. The idea, the hypothesis being that if you can inhibit or block the activation or activity of SEMA4D, you could potentially reduce some CNS inflammation and increase neuronal outgrowth in the brains of HD patients. So this study has completed recruitment, and we look forward to seeing and hearing about the results of this study. So things are moving forward, and lots of trials have come to completion and or wrapping up. So that's great news, and hopefully they'll yield good data in 2017. So just to wrap up, I threw a lot of different clinical trials at you and results, and you know, we recognize this isn't for everybody, but for those that are interested in learning more, HDSA in 2015 created a new resource, online resource called HD Trial Finder. This is basically an online matching program, free to use, doesn't require any kind of personal information to sign up and really find out what trials are available to you in your area. We did add in 2016 a call center for those who aren't comfortable with maybe placing their information online or would just prefer to talk to somebody to walk through the process and find out what's available. You can call a number, it's 866-890-6612, and talk to what we call clinical trial navigators from Monday through Friday, 9 to 5 p.m. Eastern Time, and they'll be happy to kind of walk you through the process. It's super easy. I suggest everybody kind of take a look at the site. It's hdtrialfinder.org, or you can find it on the HDSA website by clicking on Enroll in a Trial Now, and you'll be taken to this website where you just basically create an account, a profile. You just have to include a zip code for who you're searching for. Answer a short 11-question questionnaire about the HD affected individual, and you'll review your results, and your results will look something like this. This is slightly out of date because a couple of these studies are now done in recruiting, like the Pfizer and PRIDE study. But the important thing is you're going to get information about the contact in person at the site nearest you, and this is information for a New York City profile, but you'll see that if I was interested in the PRIDE study, I know the person to contact is Paula Wasserman up at Columbia, and here's her phone number, fax, and email. So just want to conclude by saying, while we've had some setbacks and it may seem like we've taken some steps back, I think that we're primed for many steps forward in the future. We should certainly have hope. As I walk through the streets of Chicago before I gave a talk at Northwestern a few months ago, I saw this shirt and it screamed at me because it was blue and the color of Huntington's and HDSA, and it certainly it just told me for sure Chicago Cubs were going to win the Super Bowl, the World Series, and they did, ending a drought of 108 years. And it brought me hope, and it kind of gave me a great peace of mind to think that there's great reason for us to have hope in all the great work that's going on in the research community. So that's all I have. So if anyone has any questions, please feel free to ask them at any point, and I'll try my best to get to them. Try to open up something here. Bear with me. Bear with me, people. I'm just trying to find the mouse. Where it went. All right. Sorry about that. Oh, did it again. Chris. The touch. Bring it back. Maybe click on that. Click on the orange. Oh, there you go. Okay, so one question is, what explains the huge drop in NIH funding? Great question. I want to know the answer as well. I was kind of shocked to see those numbers, and I know they fluctuate a little bit, but to decrease 40% had me surprised, to be honest with you. So I'll try to find that out. And is there something that the community can do to reverse that course of the NIH funding? Yeah, I mean, absolutely. You know, continue to write your senators and your Congress people to encourage them to support increased funding at the NIH. What explains the higher rate of success of the rare disease trials? So I think that's another great question, and I think that could be because there could be a number of factors there. One that I think is because, let's use Huntington's disease as an example. We know we know that the people that are in that study have Huntington's disease. So because we have a gene and we know we can do a gene test, we can be 100% certain that all the people within that HD trial have HD. In the case of Alzheimer's disease, where we've seen lots of major multi-billion-dollar phase 3 failures for Alzheimer's disease, they don't have a gene, and you know the only way technically that I'm aware of that you can diagnose Alzheimer's is post-mortem at autopsy. And we can't do that, obviously, in a clinical study. It could very well be that some of the reason that these Alzheimer's trials failed was that some of the people that were in the study didn't even have Alzheimer's to begin with. So that could explain some of it. And plus, with these rare diseases like HD, because we have a gene, we do understand and know a lot about the biology and can track these patients and know who they are and track them over time. So I think all of those could be playing a role in increasing the success rate for rare disease studies. It's a question about Enroll HD. Is Enroll HD also for people who are at risk of developing HD but do not yet exhibit symptoms? Absolutely. Enroll HD is for everyone. The entire HD, it is the study for HD families. So not only is it for both symptomatic and asymptomatic, gene-positive individuals, it's also for gene-negative family members, and a small component of it is even they even allow caregivers who are, obviously are gene negative. So it is for everybody. Why is U.S. lagging behind Europe in enrollment for studies? That's a great question, and I don't know if we have the answer for that. I don't know. I really don't know, but it could be that our doctors have maybe less time to spend with patients to really, and they're putting out fires and dealing with tweaking medicines that in their limited time that they spend with patients to really sit down and talk to them about, hey, there's a study, I was thinking about you, and there's a study that you might be interested in talking to them about it. So that could very well be part of it. It could be also just in who we are. Maybe we're less engaged than maybe the Europeans. I don't know, but we certainly do need to do a better job in improving our participation to ensure that we're not reliant on Europe for trial results, that we take this into our own hands and that we're participating in studies ourselves. Regarding the drug that was approved by the FDA in late 2016, what are the next steps? So the drug wasn't approved that I think you might be referring to. That was the SD809 drug from Teva. This is the deutetrabenazine. Actually, the FDA provided a complete response letter, which is basically saying that what they submitted was not approvable, and to provide more information. So that my understanding is what we do now is just wait, and the FDA has what they need from Teva to evaluate SD809 to see whether or not it's approvable or not. So hopefully in April we'll know more. What approaches do you get the most optimistic about? Great question. I think that's, while I don't necessarily have I get excited about a lot of things related to that slide in the central dogma of molecular biology, and that is any and all targets related to targeting Huntington. So I'm super excited, obviously, as everyone is, about the ASO approaches to lower all Huntington, total Huntington by Ionus. I think an even better approach would be to lower the mutant Huntington allele, so that I know isn't in the clinic yet, but is coming. And what would be amazing, and I know there are some companies working on this, is in the future, wouldn't it be great to be able to pop a pill that would get into your brain and lower Huntington? And I'm optimistic that the day will come that we may see something like that. So that's what gets me really excited. Regarding the drug Ionus delivered by spinal tap, what are the next steps? That's just to, as I said, that we're waiting on results in late this year to get the results from Ionus, and those will dictate what the next steps will be. If all goes well, the next step would be a phase 2 or phase 3 clinical study to investigate the efficacy of that drug in HD patients. Is China part of the Enroll program? No, not yet. But I think there are plans and hopes that maybe one day it will. Do I speak at local education days? Absolutely. I'd be happy to. What is the best course of action, in your opinion, for a person who is at risk for HD but does not want to get tested? I don't know if I can really answer that. I mean, that's such an incredibly personal decision, and either way, it's really up to you. But just know that whatever it is, folks at HDSA and locally at your centers of excellence and social workers and Support groups are all here to help and support you in that decision, whatever that decision is. A lot of questions. This is great. I'm not sure we're going to be able to get to all of them, and I know we're over time, but I'll try to get to a few more. Are there any studies currently being done among caregivers and children of parents with HD? Not that I'm aware of. There may be some surveys that are out there, and there are a number of different surveys on the HCSA website, as well as researchers around the country do put on, but there are no kind of necessarily specific interventional or observational studies that I'm aware of caregivers and children of parents with HD. I'm not going to read that one. Let's see. Which of the current studies open for enrollment has the best chance of slowing the progression of the disease? Well, I'm going to say, although it's not open for recruitment or enrollment here, I'd be lying if I said I didn't think it was the Ionus Huntington lowering study. Lots of questions. Do you think that the studying the families with late onset would help learning about HD progression and natural history? Absolutely. Yeah, I mean, this is why we want to get all of these families, both with late onset and even early onset, enrolled in studies like Enroll HD. This is where we can really learn a lot. Part of Enroll HD that I didn't talk about is there's a biorepository associated with this project where when you participate or you enroll, you give blood, and there's an option where you can give spinal fluid or you can give fibroblasts or skin cells, and those samples, those precious samples, are all archived and stored in a place. I believe it's actually in Italy, and they're available to researchers around the world to answer these types of questions and to understand why families get late onset versus those that get early onset. We need thousands and thousands of samples. You can't just answer those questions with a handful of anecdotal data points. You need thousands to make a statistically significant declaration that you think you may have identified what could be altering HD progression. So hopefully, I apologize if there were a lot of questions I couldn't get through, and I want to be cognizant of time. We're 5 minutes over, but I want to thank everyone for participating. It's been a good year, and I'm very confident that 2017 will be even greater. So thank you, everyone. Thank you for all you do on behalf of the HD community to raise awareness and funds, and just for participating in today's call. And I look forward to speaking to you again at a future HDSA research webinar series. Thank you all.